Hello, today we're going to talk about scientific investigation, a bit about how we set up scientific experiments, and we may be able to dabble a little bit in scientific theory. We'll see how far I get on this one specific podcast. Also, starting off, we have the idea of what is science. And some of the main things about science is it's supposed to be objective, and really what we mean by that is it's supposed to have no bias. This is supposed to be something that's the same for anybody, regardless of where they're at, regardless of who's doing the experiment. It's not meant to be subjective, like you might say with poetry or art, where it's different based upon who's the person that is interpreting it and their experiences. This is meant to be something that is what it is, regardless of the situation. It's something that's definite. It's something that others can check. And that comes into this idea of checking. And the idea of checking goes to self-correcting. When you're a scientist, you're going to collaborate. That means you put your stuff out there. You say, this is what I did. You can now go and you can check this, and you can see if you get the same results. If you don't get the same results, if there's some type of a problem that we find, then we have to go back and rectify it, and that's the idea of self-correction. So that's where the other scientists will work with you, and they'll sort out, all right, what is the truth? Maybe your specific experiment was flawed, maybe you just interpreted things wrong, but by checking it, we can make sure we get the right answer. And the last part of it has to do with falsifiable. And what falsifiable means is that we can prove it wrong. When you look at something in science, one of the main things that makes something science is that we can test it. And the big deal about testing is when you test something, we normally have this idea in mind that we'll talk about later, a prediction or a hypothesis, if we think we know what's going on. Sometimes you might just do an experiment to see what happens. But if you kind of think you know what's going on, you make a prediction. You're going to go through and make this hypothesis based upon what you think, based upon the research you've done, and you're going to test it. And when you test it, you can be proven wrong. You can get the results that you didn't expect. And if you get those results that you didn't expect, you are going to have to change paths. Now, you don't have to just chuck everything out the window. That's not how science works. But you can refine it. So maybe you thought this would make your blood pressure go up, and it went down. So you can revamp things and say, all right, I think this acts in the opposite way, and you can continue checking that. You were just off, perhaps. You might find it has no effect, and so your whole premise that your Flintstone vitamins will make you live forever, you just have to pitch it. It's just outright wrong. It just has no effect. That you just pay somebody money and you don't get anything in return. So there's different ways this can go, but one of the things about science is that idea of being able to test it, being able to prove it wrong. If you can't test it, it's not really scientific at that point. It can turn into science as we get more info, as we get better experiments, but at this point it's not really going to be strongly scientific because it lacks that testability, that falsifiability. Now, pseudoscience is something that gets brought up quite a bit. It's one of my guilty pleasures. Not so much that I follow and believe in it, but I enjoy listening to people talking about different pseudoscientific ideas that they have. So pseudoscience, some of the problems that you'll see with it is they try to act scientific. You'll see a lot of pseudoscientific things will have somebody in a lab coat or somebody that's a doctor telling you that this pill is going to make you live longer, or it's going to cure your depression, it's going to cure your aching joints, it's going to improve your circulation by wearing this magnetic bracelet. But for all they try to act scientific, they're not scientific, and that can be for a variety of reasons. The first one is sometimes they're not falsifiable. They are going to go through and they are going to make claims that we really can't verify. So they might say that there's this hidden energy that's going to make you feel better, but it's not detectable by any modern current machinery. If we can't test it, if we can't verify it, it's not scientific. There's no way of knowing if it's even there. There's no way of knowing if it's what's causing the effect. So at that point, it's just someone's idea. It's not scientific. The other thing is oftentimes they'll just be not tested. Someone will just say, I think vitamins will make you live longer, or I think vitamins will cure illness or something, but there's nothing to back that up. There's no study where they gave people vitamins and checked that. And so initially when they make these claims, which may sound good, vitamins are good for you, take vitamins and you'll be healthier. But ultimately that doesn't necessarily mean that it's true. You'll see a lot of times too, there's these vague claims, like you'll be healthier. What does that mean? And so that can also be where it's hard to falsify because you don't really know what you're testing. You don't know what they're referring to. I'm going to kind of jump just because of this. And then a lot of times you'll find there's actually evidence against. So like for vitamins, unless you have a horrible diet, which is pretty much somebody who's probably going to be in at least what they used to call third world country. It's usually called developing now, although I think that's changed too. But if you're in certain Latin American or African countries where there's not enough food, the food quality can be very poor, vitamins may help you. But for the vast majority of people that have an okay diet, vitamins don't really do anything for you. It's kind of like going to the store and trying to buy something for $10 just because you have $12 doesn't mean that it really affects that transaction. You just have extra. So in general, you'll see that oftentimes there are experiments that are done, and the experiments actually are against what the pseudoscience is trying to say, but they'll ignore them. Or they'll pick and choose data, oftentimes improperly. And so this is one of my favorites because they oftentimes use things like quantum mechanics or quantum physics, which if they bring that up, it's pretty much a guarantee that they don't know what they're talking about. They'll look at something and say, well, quantum science has proven that there's these irregular things that happen, but those irregular things happen in specific situations that are irrelevant to what they're talking about. And so oftentimes they'll try to grab these things that are scientific but have nothing to do with what they're trying to convey, and that way it tries to lend credibility to them. So you just need to be mindful about pseudoscientific things because they can be very tricky because they have the trappings, they have the looks of science. So really, if someone seems to be pushing that something's scientific, you should probably start to seriously doubt whether or not it's actually scientific. If they really have the person dressed up and they keep emphasizing who exactly, like the doctor or PhD, there's a good chance that they're trying to just take your money. Okay, moving on to kind of what scientists do. One of the major things they do is make observations. And an observation is just something that we can pick up with our senses, although it doesn't have to be our bare senses. So certainly I can say I heard something, I can say I touched and I can see the feel of my computer, the keys and the keyboard. I can look and I can see that there are letters on the keys and the keyboard. Those are my direct senses. But you can also have technology aid us, like microscopes and telescopes. I can't, squint and see all the details on the moon. There's certain things that are beyond our senses unaided. But that's okay because we can use aid devices. And so that's going to be things, like we said, like microscopes, telescopes. We have certain sensors and stuff that can tell us like the amount of radiation, which we can't see elsewise. There's lots of stuff like radio waves that you can pick up and you can detect and you can decode. You can listen to the radio, but we can't like look around us and see radio waves. And so we'll use other tools to allow us to observe them because now I can see the needle moving in a certain pattern, or I can see that the computer gives me a specific code for the size of the wavelength. And so that allows us then to understand characteristics directly from our observation, but only because we had that technology. And inference is very similar. I just want to bring this up because a lot of times we can't directly observe something. And so instead we can infer it based upon what we can observe. So we observe essentially the effects, and so we can infer the cause. Now sometimes you have to be careful because if you don't have good enough observations after, good enough effects if you will, you can't really have much certainty of the cause. So you've got to be careful to make sure there's enough evidence that you have as far as the observation, the effects, to make sure you can properly determine the cause. So don't just sit there and say like, my grandfather ate a lot of peas, he lived to be 100, therefore peas will make you live to be 100 years old. Uh, you don't have enough evidence to show that everybody who's eating peas is living these long lives. You have one specific case, and there's lots of other stuff that could have caused that. Whereas there are other situations where you might see where it hasn't rained in days and days, and you know that, you've observed that, and then suddenly the dog comes in from outside where there's no pool, there's no source of water really other than the hose, and it comes in wet. You might also detect that there's a scent of like soap on him. So you could infer he got a bath, and that'd be a pretty solid inference. And that's the difference, though, is you have pretty concrete evidence based upon the observations that are around there to determine this. So that's going to be an inference. So this picture is meant to kind of get you guys thinking about this. So we've got a small set of footprints that's coming in through here, up at the top. You've got a larger set of what looks like dinosaur or bird footprints that are coming in at an angle towards the smaller set from the bottom, so it looks like they're converging. So I could look at this and say these animals are moving to the same general area. One of them appears to have bigger feet, the other one has smaller, longer feet, and then I could probably make some inferences, like the one with bigger feet is probably larger than the one that has these smaller feet overall in size. And then depending, I could infer maybe that they're going to meet, but that could be that there's different times that these occurred. They could have fossilized. So it could be that they didn't meet. They just walked across the same spot, but they did so hours or days or years apart, depending on exactly what those footprints represent. So then you can continue. You'll notice at the end, though, it does have where the bigger one, the one with bigger feet, the feet do get spread out a little bit more, which one might infer would be like running. And so then when we look at the next thing, we can see that they do meet up. The one keeps spreading out further and further, the large footprints, which makes me once again think it's probably running to the other. And then you see this kind of big ball of just both of their footprints mixed off to the far right, where it appears that they did meet physically and something happened that they went around and around. Two options that I could infer is perhaps it was something reproductive, or perhaps one's going to eat the other. I think that there's a stronger reason to believe they'll probably eat one another just because they don't have similar footprints. So you usually don't mate with somebody else who's not like you unless you're really desperate. So in this case, I'm assuming not. And then if you see the last one, it does show that only the large set of footprints go away. So we see that the small footprints are now gone, so there's a good chance that they're gone. We can infer pretty solidly because it's dead. It didn't walk anymore. It was done. And so overall, we can get a better and better picture as we get more and more evidence and we get more and more concrete about what's going to happen. And then we'll discuss this one a little bit in class, but just to think about it, it's a picture of a squirrel with a zipper in the center, and its little hands are pulling the zipper apart and showing a Superman symbol. If you're listening to just the audio, I'm trying to verbally describe it just so that way they can still know what's going on. And so it wants you to list 3 observations and 3 inferences. So if you were to see a picture of what looks like a relatively normal squirrel, except it's pulling apart a zipper on its chest to expose a Superman logo, you can think about what your observations would be and what your inferences would be, and we'll discuss that in class. Okay, next is the scientific method, which isn't an exact thing, but in general, scientists are going to do a lot of these general characteristics, general traits, general steps. So we're just going to kind of cover the major things. So in general, when scientists are studying something, they're going to have something they're curious about, they're going to have a question, they're going to have a problem, and they're going to research it, they're going to study it, they're going to find out as much as they can about it, because that's one of the first steps when you're studying something is figure out what we know. And by we, I mean the scientific community. You're not the first person necessarily to look at it, so you want to see what else has been done prior. From there, if you kind of have an idea of what's going on, you can construct your hypothesis. All right, so you make your prediction. You then are going to go through and you're going to test that hypothesis by designing and performing an experiment. Once you do that, you're going to analyze the results. So that means you're going to look at those results, and then you're going to conclude or decide whether or not your hypothesis was valid or not. That's one of the major things you're after. And if it's either way, actually, you're going to report what you did, because even if you find out that you were wrong, it still allows other scientists to build on the fact that the hypothesis you had was wrong. And so that allows other scientists, as well as you, to now go back in many cases, if you were wrong, to redo it, to change the hypothesis and try again to try to get the right answer. If you find out that you were right, you would typically repeat it anyway to verify, and other people would typically repeat it to try and verify as well, because you don't want to just get one result and go, oh yeah, I'm done. Jurassic Park, the movie, came about because a guy accidentally contaminated a sample when he drew blood out of insects that were stuck in like tree sap, amber they call it, and he contaminated it with a piece of his turkey sandwich. So when he got DNA from that seemingly long dead insect that likely would have been from a dinosaur it fed on, there was this big idea that Jurassic Park, we can get DNA from these insects stuck in amber. When in reality, when other people tested it, they didn't find that. And then when they eventually tested the DNA they found it was from turkey. And I mean a turkey, not the country or anything else, but ultimately just from a regular old turkey. So that was a contamination, and so that whole thing spawned a movie series and all this stuff. Ultimately was just a mistake, but science caught that because it checked. And that's why it's so important at the bottom here that pretty much no matter what you do, you report your results. It's okay in science to have your hypothesis be wrong. That can tell us just as much as if it was supported. It lets us move on and try again. Now, for experimental design, I'm just going to try to go through some of the quick things. First of all, you have to realize how many variables you can test at a time. And that one's a pretty simple one. You are allowed in any scientific experiment to test one variable at a time. And the reason for this is if you change two things and you find out you get the result that you wanted, we don't know which one fixed it. We don't know which one caused it. Chances are only one of those variables was important. By changing two of them, I don't know which was the right one. So we always change one variable at a time, and as we eliminate variables that don't actually make things better, that's why I said it's so valuable, because we can move on to the next one until hopefully we find one that works. Or maybe we find none of them works and you scrap that kind of idea, it didn't pan out, and you can move on to new ideas. Or you can look at the problem in a different way and try to go about solving it in a different way completely. And then below this I've got the two groups, control group and experimental group. The control group is one that you basically leave unchanged. The goal here is we want this for comparison, all right? We want to know if we think coffee is going to kill you. We want to have somebody who's not drinking any coffee so we can see, all right, when did they die on average? So we can then compare that to the experimental group, which are the ones that we manipulate, where maybe we have some of them drink 1 cup of coffee a day for their life, 2 cups of coffee, 3 cups of coffee, 4 cups of coffee. So we can then compare how coffee affected their lifespan, and we can compare that, the people who drank coffee of different amounts, their lifespans, to not drinking coffee at all. Because it could be that any amount of coffee is dangerous, or it could be that some coffee is good, maybe 1 cup a day is better than none at all, but 4 cups is bad for you. And so you need to make sure that you don't just give everybody coffee and then you say, well, 1 cup of coffee is the best thing you can do, when in reality any coffee is dangerous. I don't know if that's true, so don't hold me to that. I'm just giving you an example. But that's the idea of what can happen. And so the experimental group is the ones that we actually manipulate, is kind of how I'll say it. So we're going to actually expose them to changing variables. Whereas the other guys, we just kind of leave them be. In a medical experiment, the control group is usually given a sugar pill called a placebo. They're not given the actual medication. And they do this to make sure that people don't just go, oh yeah, I feel less pain, because they expect to feel less pain, what they call the placebo effect, and that it's actually the medication. So it's important to have a control group, especially with human subjects, because we can kind of mentally delude ourselves into certain things, where a lot of times if you think something's going to happen, your brain can kind of make that happen. Now this doesn't mean you can fly or something, but when it's things just like levels of pain, stuff like that, you can oftentimes improve things just by giving somebody this placebo, just making them feel like you're going to make them feel better. And then variables, other variables, the constants are all the other variables in the experiment. So if we're doing like coffee, we would want their diets, we would want the amount they work, and all the other things that they do, we'd want those to stay as close as possible to the same. And this is the idea that we only want one variable to change. And so because we only want one to change, the others need to stay the same. So I'm just going to write one change only, because we only want one variable to change. The rest of them need to be the same. The independent variable, which I'll just write as IV in the future here, is the ones that we change. This is the one that, as an experimenter, you say, drink 1 cup of coffee, drink no coffee, drink 2 cups of coffee a day, drink 3 cups of coffee a day. That's what you're controlling. So the independent variable is the one that's under your control. It's the one you can manipulate. Sometimes, too, you might see it's called the cause. This is what we're changing, and we're doing that to measure the dependent variable. So this is where we're trying to see the effect of our cause. So if this was ultimately coffee, this down here might be something, the effect would be something perhaps like lifespan. How long did you live based upon the amount of coffee that we gave you? That's what we're after. So we'll talk a little bit more about those later, but in general, just independent variable, you control. Dependent variable, this is what you're trying to measure. You're trying to see how it's affected by what you changed. And then a hypothesis, this is just going to be a statement that we make as scientists that's a prediction. It's testable, so it means we can check it. And it's oftentimes, and for our purposes, we'll pretty much always do it in an if-then statement. So this is going to be something we are going to test to see if we're right, and we will write if we do something. So if the temperature of the room is increased, so I'm controlling temperature, and because I'm controlling the temperature, I'm cranking up the thermostat for each different sample. That's going to be our independent variable. Then the bacteria will grow faster. So this is what we're expecting to be measured. So we're assuming that the higher the temperature, the more bacteria that we'll get. And so this bottom part is basically our prediction for what will happen to our dependent variable. So when you set up a hypothesis in this if-then statement kind of way, it makes it pretty easy to make sure that you're doing what you're supposed to, which is to make this prediction about when I change this specific something, the independent variable, in a certain way, this is what's going to be measured, this is what's going to happen because of that. So if you look at some of these other examples, if you spend more time studying, then your grade will improve. So you guys at this point should be able to figure out what's my independent variable, what's my dependent variable. So if you spend more time studying, then your grade will improve. You guys should be able to sort that out. Any other statement that I give you that's an if-then statement, you guys should know which variable is which, what's going on. What are you controlling, what are you measuring, and what are you supposed to measure if it's correct? The hypothesis, I mean. So I hope you guys have enjoyed this, and I will catch you guys in the next podcast.